alluded to a lot while discussing the acid-base properties of our amino acids was that amino acids are usually incorporated into other structures. Generally, amino acids get incorporated into proteins because we have a lot of proteins in our cells as well as in our bloodstream and other places within our body. And these are all made from amino acids as kind of the building block. And the first thing that has to happen is that two amino acids have to come together and make a peptide bond. And that comes from the reaction of a carboxylic acid with an amine. And since all amino acids have an amine and a carboxylic acid, basically we can make polymers, fairly large polymers actually, if we want to make a large protein. We can string together hundreds of amino acids to make some fairly large proteins. Basically what we see is that carboxylic acid here reacts with the amine that we see here, and the result is a loss of water, loss of H2O, and as a result we end up with, this is the carbon that came from the carboxylic acid, and here's the nitrogen that came from our amine. Now, the other thing that we lose is the negative charge that we had on the carboxyl group and the positive charge that we had on the amine. The amine group, the nitrogen that we see here, these lone pair are not available to pick up a proton as they were with the free amino acid. So we can't protonate this, we don't get a positive charge on this functional group. So as a result, the amide bond that we make is always going to remain neutral under typical conditions within our body. So we lose a little bit of charge, but not that big of a deal. What we create from this is basically larger structures. So if we put two amino acids together, we form what's called a dipeptide. Stick another amino acid on that, we end up with a tripeptide. As we keep adding amino acids, eventually we make something large enough that we don't really call it a peptide anymore, we call it a protein. And proteins play extremely important roles within our cells. Basically, our cells would not function without proteins. So the first making peptides and then progressing on to making proteins is an important thing that gives rise to the ability for our cells to function to make a living organism. So we'll talk a lot more about some of the properties of the peptide bond in the next chapter, but for now we're going to talk about some of the peptides, some of the basic peptides that we see that play roles within our cells and within our body as a whole. So I'm going to show you a variety of different peptides that we find in nature. We'll start with a class of molecules called the enkephalins, and these are molecules that we produce naturally. They actually bind to the opioid receptor, and we had no idea what enkephalins were, but we knew what opioids did. Opioids generally, relieved pain and made one feel pretty good about life. So eventually, from knowing that we had this receptor for molecules that were derived largely from plants, we're kind of wondering, well, why do we actually have this receptor for something that we do not produce in our body, and we really don't find it in very large amounts in nature. So basically that led eventually to the discovery of molecules such as the enkephalins. And enkephalins are short peptides. If we look at the structure here, the first amino acid, if we look for the free amine group, so there's the start of this peptide. Here's the first alpha carbon, and then here's our first carbonyl that came from a carboxylic acid. This amino acid here would be tyrosine. Our next amino acid starts right here, and here's its alpha carbon. The alpha carbon here has just two hydrogens, so this would be glycine, and then there's the carboxylic acid from that. Here is our next amino acid. Here's the alpha carbon. Once again, two hydrogens, so it's once again glycine. Here's our next amino acid, and if we look at this structure here, this gives rise to phenylalanine. And then our last amino acid here basically here's our alpha carbon, and the coming off here, this R group here is actually going to be the amino acid leucine. So this is what we actually refer to it as a Leu enkephalin, to indicate that leucine is found at as the last amino acid, the amino acid at the carboxy terminus of this peptide. So the other class of peptides that behaves a lot like the enkephalins are molecules called endorphins. And those of you who are runners have probably heard of endorphins and have had maybe that endorphin high. Enkephalins do kind of a similar thing. They work in combination with endorphins and are also released along with endorphins. So they have some very similar physiological properties. So I've got this molecule listed as a not so fun peptide. This is aspartame, better known as NutraSweet. We find it in foods that generally want to have relatively low calories but be sweet. So things like most of our diet sodas actually contain aspartame. The function of aspartame is as a sugar replacement. It basically binds to a receptor in our mouth that is referred to as the sweetness receptor. And as the name implies, it obviously detects sweetness in our foods. They were not using much imagination when they named that receptor, obviously. But aspartame happens to bind to the sweetness receptor about 200 times more tightly than sugar does. So as a result, we can use much less of this molecule than we would have to use of typical sugars. So for example, most of your soft drinks contain about, one can contains about 40 grams of sugar. But you can replace 40 grams of various sugars with about maybe half a gram or so of aspartame. And when I say sugar, what I really mean is high fructose corn sweetener. That's what is very often found in most of our soft drinks these days. So anyway, so aspartame, if we look at the structure of aspartame, here would be the N-terminus. And we've got, this is the first alpha carbon. Coming off here would be aspartic acid. We then have our next amino acid. Here's our alpha carbon, and then the R group here makes this phenylalanine. And then this is actually a methyl ester. The addition of that methyl group onto that carboxylic acid makes this bind to the sweetness receptor much more effectively than would a free negative charge. And that's simply due to the structure of the sweetness receptor. It can accommodate the negative charge that we see up here, but it cannot really accommodate a negative charge on this carboxylic acid. So having that methyl group there is actually really helps this molecule give rise to its ability to bind to that sweetness receptor. So because there are two amino acids in it, aspartame would be considered a dipeptide composed of two amino acids. And we'll see aspartame later on in this course when we talk about various other aspects of food biochemistry. So I've got oxytocin here as kind of an awesome peptide. Oxytocin is very interesting, a very interesting structure. It's responsible for a lot of things that happen in our body, particularly during motherhood. This is oxytocin is what actually stimulates uterine contractions to basically start the birth process. It's also involved in breastfeeding and many other events that help a mother and a baby bond to each other. So if we look at the structure of oxytocin, basically one of the things that we see in this is the fact that we have, this is a cysteine residue here. Here's the here's the amine group, here's the alpha carbon, here's the carboxylic acid, and then here's the R group. And that R group contains a thiol group, but we'll notice that that thiol group is actually bonded to another thiol group that comes from another cysteine residue. This is what's referred to as a disulfide bond. And two cysteine residues frequently are, if they're in the right conformation within a protein, or in this case a peptide, they're able to react with each other, and they undergo an oxidation reaction, and as a result they actually form this sulfur to sulfur bond. This is actually very important for a lot of aspects of protein structure. In fact, it's one of the things that gives rise to the structure that we see in our hair. It's what part of what makes hair curly versus straight. And we can change our hair basically by simply reducing the disulfide bonds that are present, and then changing whether our hair is straight or curly, and then reoxidizing those cysteine bonds, or cysteines, to form the disulfide bonds that we see here. Now, generally, what one can do is one can, have, with straight hair, they can go to curly hair by essentially reducing the disulfide bond and kind of rearranging the disulfide bond pattern, and then adding an oxidizing agent to reoxidize those thiol groups to reform that disulfide bond. So basically, the chemistry behind getting a perm relies upon the same bond that we see in the molecule oxytocin. So, and what it does in this case is it actually gives the oxytocin structure a somewhat cyclic structure. We would call this portion right here is essentially a cyclic portion of the peptide, and the rest of this is just a kind of the rest of the peptide tail hanging off there. So we can also do a variety of reactions with amino acids, and a great example starts with the amino acid tyrosine. And tyrosine, basically, if we use the enzyme tyrosine hydroxylase, it puts a hydroxyl group on this position to give us what's called dihydroxyphenylalanine, or L-DOPA, as it's usually known. And then the L-DOPA is then reacted upon by dopa decarboxylase to lose this carboxylic acid, and this gives rise to dopamine, which is a neurotransmitter that we have. And dopamine is responsible for basically, the initial spark you get when you think you like somebody. Dopamine's that thing that makes you feel silly and stuff like that. But anyways, so dopamine and oxytocin both are involved in feelings of bonding between people. They just play very different roles. Dopamine is more of a short term, whereas oxytocin is something that we feel for long-term attraction between people. And like a long-term attraction between a mother and a child is largely brought about by oxytocin. So as a fun little chemistry thing for the end of this chapter, there's a lady who lives in Hawaii who actually got a PhD at UC Berkeley in biochemistry. She did a lot of research on gene editing. Some of the basics of gene editing came out of the lab she worked in. And after getting her PhD, she decided that she didn't want to really do research anymore. So instead, she started making silver jewelry that depicted well-known molecules. And a couple examples are she basically made dopamine, which you see on the left, and oxytocin, which you see on the right. And these are basically, they're beautifully done, and if you're interested in, giving a nice gift to the significant other in your life, these are excellent ideas for you. And she has a website called Made with Molecules that basically showcases all the jewelry she does. And anyways, if you're interested in the transformation of chemistry into art, and in this case into some really nice jewelry, it's kind of a neat little site to check out. So anyways, that's it for amino acids. Our next chapter will look at protein structure.